Good afternoon, everyone. This is Rosemary Shaga, and we are Group 3, and we will discuss week 7 to 8. So, we are now starting the discussion. So, week 7 to 8, unit learning outcome. ULO 1, explain the metabolic pathways in its intermediate compounds. ULO 2, explain the amount of energy produced during the oxidation of fat. Essential knowledge. Certain classes of lipids play an extremely important role in cellular metabolism because they represent an energy-rich fuel that can be stored in large amounts in the adipose or fat tissue. Between one-third and one-half of the calories present in the diet of the average U.S. resident are supplied by lipids. Furthermore, excess energy derived from carbohydrates and proteins beyond normal daily needs is stored in lipid molecules in adipose tissue to be mobilized later and used when needed. Lipid metabolism. Digestion and absorption of lipid. Dietary lipids, about 98%, triacylglycerols or TAGs, fats, and oils. Salivary enzymes are water soluble in the mouth have no effect on lipids or TAGs, which are water insoluble. In stomach, most not all of TAGs change physically to small globules or droplets called chyme, which floats above other material. So this will be the diagram. The dietary triglycerols or TAGs, first mouth, has saliva no effect on digestion. When we eat some fat molecules, an enzyme called lingual lipase is released in saliva in the mouth. However, this enzyme is not actually activated in the mouth. Next, the fat molecules in the saliva with the lingual lipase enzymes is swallowed and enters the stomach. Here's the second. Stomach churning action produces small fat droplets or chyme. Second is the gastric lipases or hydrolyze some 10% of TAGs. So the stomach, the highly acidic environment of the stomach, activates the lingual lipase enzymes, which are also joined by gastric lipase, which are enzymes released by chief cells in the stomach. These enzymes begin the digestion of fat molecules by breaking some of them into the glyceride and a fatty acid. These lipases act on about 30% of triglycerides. So the next will be the small intestines. Next, the fat molecules enter the small intestines from the stomach. Because the fat molecules are hydrophobic or water-fearing, they enter into the small intestine as large fat or lipid masses. So the body must first break down the larger masses into smaller droplets, which is called emulsification. Now the pancreatic lipases, which is an enzyme released from the pancreas into the small intestine, has greater access to the fat and can digest or break them down into mainly monoglycerides and free fatty acids. This then associate with the bile salts and phospholipids from micelles, which are much smaller than emulsions droplets. And that will be the bloodstream, that have the free fatty acids in bloodstream. The TAGs are hydrolyzed to free fatty acid. So that then transported through the lymphatic vessels and eventually enter blood, where they are delivered to the liver, fat tissue, muscle tissue, and to other tissues as well, where they can be stored or used by cells. And next. So next slide, lipid digestion starts in the stomach. A triglyceride consists of a glycerol unit and three fatty acid chains. Triglyceride molecules are non-polar molecules, meaning They don't mix with water, so they are hydrophobic or water-free, as to speak. And the fat molecule, the fatty acids are attached to each of the three carbons of the glycerol molecule with an ester bond through the oxygen atom. During the ester bond formation, three molecules are released. Since fats consist of three fatty acids and glycerol, they also called triglycerols or triglycerides. And monoacylglycerol lipase catalyzes a chemical reaction that uses water molecules to break the glycerol monoesters for a long chain fatty acids. So that would be my topic.
Glycerol metabolism. Taken to liver or kidney by blood converted to dihydroxyacetone phosphate. Recall that DHAP is part of the glycolysis pathway. This compound may be converted to lactic acid or to glycogen in the liver or muscle tissue, or to pyruvic acid, which enters the TCA cycle. Thus, the glycerol part of a fat is metabolized through the carbohydrate sequence. So these are the sequence right here. Glycerol metabolism. In glycerol metabolism, the liver and kidneys are primary organs that metabolize serum glycerol. Glycerol kinase or GK catalyzes the conversion of glycerol into G3P, which can be used for lipid synthesis or enters the glycolytic pathway after being oxidized into DHAP by FAD-dependent GPDH. Oxidation of fatty acids. So there are three parts to the process by which fatty acids are broken down to obtain energy. So the first part is the activation, and the second one is transport, and the third one is repeated oxidation. So for the fatty acid activation and transport, activation of fatty acid takes place in outer mitochondrial membrane. Fa reacts with coenzyme at the expense of 2 moles ATP to produce high energy acyl coenzyme A. Coenzyme A is too large to pass through the inner mitochondrial membrane to the mitochondrial matrix, where the enzymes needed for fatty acid oxidation are located. It is converted to acyl-carnitine, a shuttle mechanism involving the molecule carnitine affects the transport of acyl CoA into the mitochondrial matrix. So for the transport, it is a long chain fatty acid transport across the cell membrane has long been thought to occur via passive diffusion. However, there has been a fundamental shift in understanding in recent years, and it is now widely accepted that fatty acids cross the cell membrane via a protein-mediated mechanism. And for the repeated oxidation, fatty acid oxidation is the mitochondrial aerobic process of breaking down a fatty acid into acetyl CoA units. These are the sequence of the fatty acid activation and transport.